Welcome all of you. I am Rosalind Mishra. Welcome to our class of Pharmaceutical Inorganic Chemistry. Okay, so in today's class we shall be discussing about our topic Hematinics. Okay, now so before entering into Hematinics, let us first grab a little introduction about our lecture. So I shall be dealing with Pharmaceutical Inorganic Chemistry in our class, which is this unit is our unit 4 according to our syllabus. So unit 4 deals with various topics like expectorants, which we had discussed in our previous class, emetics, again we had already finished discussing about emetics. Then we started with hematinics. In our just previous class we had discussed about various facets of hematinics, what hematinics are, why are they needed, for treatment or of what disease do we use these drugs known as hematinics. Okay, and apart from this, as we can see here in our slide, our syllabus expects us to learn two major types of hematinics, which is ferrous sulphate and ferrous gluconate. Ferrous sulphate and ferrous gluconate. See, in our previous class already we had discussed about ferrous sulphate. We have seen the structure of ferrous sulphate, we had seen the physical properties of ferrous sulphate in details, we had discussed the chemical properties of ferrous sulphate in details. Apart from that, we have also discussed about the assay of ferrous sulphate, various uses of ferrous sulfates, etc. In our today's class, we shall be moving on towards ferrous gluconate. We shall be discussing about ferrous gluconate in detailed manner. Okay. Now, before learning about ferrous gluconate, first let us see what is this hematinics all about. Okay. So, since today is our fresh class, so we shall be first learning a little bit about hematinics, and then we will move on to learn about ferrous gluconate. We shall see the structure of ferrous gluconate, their physical and chemical properties, their synthetic procedures and uses of ferrous gluconate. So, in our today's class, we shall be learning about hematinics again in detail, and then we will move on towards a very important hematinic, which is ferrous gluconate. So, now what is hematinics? Since a long time, since I guess around last 2 minutes, we have been talking about hematinics. What is this term hematinics? What is meant by hematinics? What are hematinics? Hematinics are the agents used for formation of blood. Clear all of you? What are hematinics? These are the agents which are used for the formation of blood to treat various types of anemia. Hematinics are used for treatment of anemia. Hematinics, heme, yani blood. They are used for formation of blood in order to treat various types of anemias. Okay, so few examples of hematinics can be underlined as iron, vitamin B12 and folic acid. Okay, now what is this anemia then? अभी हमने hematinics का तो जाना, that hematinics are used for treating anemia. ये anemia क्या होता है? What is meant by anemia? Well, anemia is a condition. It is a condition which is highlighted by 3 major categories, or anemia is a condition which is defined by 3 major things. Number 1, anemia is defined by the decreased capacity of RBC to carry oxygen to tissues. Anemia is defined by the decreased capacity of the RBC to carry oxygen to the tissues. Now, what does RBC do? What is the major role of RBC? What is RBC composed of? Well, I shall answer all these questions one by one. Number 1, RBC is the major component or constituent of blood. What is blood? Blood is the major fluid in a living system. Blood is the fluid in the living system. Blood is composed of RBC. RBC contains hemoglobin. RBC contains hemoglobin. What is this hemoglobin? See, human body contains blood. Okay, blood contains RBC. RBC contains hemoglobin. RBC contains hemoglobin. This hemoglobin is a protoporphyrin. It is a protoporphyrin structure. It carries iron molecule. This hemoglobin carries 4 iron molecule. One molecule of hemoglobin can carry 4 molecules of iron. This iron carries the oxygen from our lungs to the various tissues in our body. They help in transfer of oxygen. They help in the transfer of oxygen from lungs to the tissues. From lungs to various tissues in our body. Okay. So what does this do? RBC do? They contain hemoglobin. These hemoglobin carry oxygen from lungs to distant tissues in our body. In the tissue, they exchange this oxygen. They exchange the oxygen with CO2. They exchange the oxygen with CO2 and carries back this CO2 to the lungs so that it can be exhaled out. It carries back the CO2 back to the lungs so that it can be exhaled out. Okay. So this is the role of RBC. If this RBC's activity or if this RBC's role to carry oxygen to the tissues gets hampered, then it leads to the condition called anemia. Okay. This hampering of the capacity of RBC to carry oxygen or the oxygen carrying capacity of the RBC, if this gets hampered, it leads to the condition called anemia. Okay. So now let's move on to the next reason for anemia. The other reason due to which anemia can be caused is when the balance between production and destruction of RBC is hampered. Anemia can also be caused when the balance between production and destruction of RBC gets hampered. See, RBC has a life of 120 days. RBC has a life of 120 days. In 120 days, these are known as functional RBCs. Functional RBC are the ones that has got very good oxygen carrying ability. Okay. They help in the transfer of oxygen and they help in the exchange of carbon dioxide. These functional RBCs have a lifespan of 120 days. Within 120 days, the functional RBCs get destroyed, new RBCs are formed. It is a cycle. It is a very healthy and balanced cycle. If there is a destruction in this cycle or if there is a disturbance in this cycle, then it leads to the condition called anemia. Then it leads to the condition called anemia. That is, when the synthesis of RBC is lesser than the RBC which is getting destroyed, then it leads to the condition called anemia. Clear all of you? So, what are the reasons for the disturbance in this cycle? RBC produced and RBC destroyed. RBC destroyed. This is a cycle. Basically, it is a cycle. If the balance or if this cycle is hampered, then it leads to anemia. What are the reasons for the hampering of this cycle? वो क्या reason है which could lead to an imbalance in this cycle? They are blood loss. They are blood loss. If a person suffers acute blood loss due to whatever might be the reason, then it disturbs this functional RBC number, disturbs this functional RBC cycle. Hence, it leads to anemia. Thus, it leads to anemia. Now, other reasons. There are various other reasons for this imbalance of cycle. They are impaired red cell formation. Impaired red cell formation. What are the reasons for this impaired red cells formation? They are deficiency of essential factors, that is iron, vitamin B12 and folic acid. See, as I told you, the destruction of RBC is followed by production of new RBC. If this production of new RBC is hampered, it will lead to shortage of RBC count. In what are those cases in which this production of RBC is getting hampered? Deficiency of essential factors. They are deficiency of essential factors like folic acid, vitamin B12 and iron. These are the factors whose deficiency might lead to decrease in the RBC count. Clear all of you? Now, what are the other reasons for decreased formation of RBC? They are bone marrow depression. Bone marrow depression. Also known as hypoplastic anemia. Bone marrow depression, also known as hypoplastic anemia. Also known as erythropoietin deficiency. Another reason is erythropoietin deficiency. Erythropoiesis is the genesis or the synthesis of erythrocytes. So, erythropoietin deficiency leads to decreased erythrocytes. So, in this what have we learned? Here we were looking into the reasons for anemia. The first reason is RBC's count gets reduced due to sudden loss of blood. The other reasons are RBC counts getting reduced due to impaired red cell formation. Due to improper formation of RBC, the RBC count gets reduced. What are the reasons for this improper formation of RBC? The reasons are deficiency of essential factors like iron, vitamin B12 and vitamin B6. These factors are essential for synthesis of RBC. The other reasons being bone marrow depression. Bone marrow depression is also known as hypoplastic anemia. Bone marrow depression is also known as hypoplastic anemia. Another reason for impaired red cell formation is erythropoietin deficiency. Erythropoietin deficiency is another major reason for impaired red cell formation. Now, other reasons for anemia. First, we have seen sudden blood loss. Second, we have seen impaired red cell formation. The third reason for anemia could be increased destruction of RBC. Increased destruction of RBC. This condition is also known as hemolytic anemia. Increased destruction of RBC is also known as hemolytic anemia. That means the RBC in our body starts getting destructed. They get deformed or destructed. Iron deficiency occurs due to the following reason. Okay, they are malnutrition, loss of iron, congenital, a condition called congenital atransferenemia. A condition called congenital atransferenemia. What is meant by this atransferenemia? It is the inability to release iron from transferrin molecule. That means iron is present in our body, but free or the elemental form of iron is not available for activity. This condition is known as congenital atransferenemia. Now moving on further. Here we could see the various classifications of anemia. Various classifications of anemia. What are the different types of anemia? Number one, microcytic anemia, also known as or microcytic hypochromic anemia. It is mainly caused due to iron deficiency. It is mainly caused due to iron deficiency. The next form of anemia is macrocytic anemia. This is also known as megaloblastic anemia. Macrocytic anemia, also known as megaloblastic anemia. This is mainly caused due to the deficiency of vitamin B12 and folic acid. It is mainly caused due to deficiency of vitamin B12 and folic acid. What is meant by macrocytic anemia? What is meant by megaloblastic anemia? Well, this is the condition where the size of RBC is increased. There is an increase in the size of RBC. This increase in the size of RBC leads to decrease in the count of RBC. Okay, decrease in the count of RBC. Hence, the oxygen carrying capacity of the RBC gets reduced. Due to larger size, due to increased size of RBC, the oxygen carrying capacity is reduced. Oxygen carrying capacity is reduced. Okay, because there is a reduction in oxygen carrying capacity, hence it leads to anemia. Our body does not transfer oxygen, or it fails to transfer adequate amount of oxygen to our distant tissues. Hence it leads to anemia, or this condition is known as anemia. Clear all of you? Now, increased size leads to decrease in oxygen carrying ability. Apart from this, due to the increase in size, another condition occurs, that is, the number of functional RBC also gets decreased. Functional RBC number gets decreased. There is a decrease in the number of functional RBC. Hence, again hampering the oxygen carrying capacity of the blood. The next kind of anemia is hemolytic anemia, where there is destruction of RBC, where there is disruption or destruction of RBC. Hemolytic anemia. Another type of anemia is pernicious anemia. Pernicious anemia. Pernicious anemia sees a decrease in the number of intrinsic factors. Intrinsic factors, also known as IF factors. Okay. These decrease in intrinsic factors leads to non-formation or decreased formation of RBC, thus leading to the condition called anemia. Okay. So, what are the different types of anemia we have discussed? These are the commonly known types. वैसे there are a lot of types of anemia known, but the commonly known types of anemia include microcytic anemia, microcytic hyperchromic anemia, the second one macrocytic or megaloblastic anemia, the third type known as hemolytic anemia, and the fourth type being pernicious anemia. So, these are the different types of anemia in which there is impaired function of red blood corpuscles. These are mainly caused due to decreased RBC formation or decreased capacity of RBC to carry oxygen. Now, in all these conditions, we have seen iron is the major factor. We have seen iron playing the major role. So, let us first understand how is this iron available in our living system. We will discuss the availability of iron in our living system. Iron is a vital body constituent. It is a vital body constituent. Total body iron in an adult is 2.52. It's in the range of 2.5 and 5. Now, what are the forms in which iron is available? Do you really believe that whatever iron we take in, it is available for our body? No. The available form of iron is in these forms only. Okay. That is, iron is available for the functionalities in our body in the form of hemoglobin. 66% of iron is present in our body in the form of hemoglobin. Okay. Iron is also stored as ferritin. Iron is also stored as ferritin and hemosiderin at a range of 25%. Okay. So, 66% of iron is present in the form of hemoglobin. 25% of iron is present in our body in the form of ferritin and hemosiderin. In the form of ferritin and hemosiderin. Iron is also present in our body in the form of myoglobin in our muscles. In the form of myoglobin. It is present in our muscles. 3% of iron is present as myoglobin in our muscles. In enzymes, iron is present as parenchymal iron. In our enzymes, iron is present in the form of parenchymal iron, ranging up to 6%. Ranging up to 6%. Okay. So, now students, till this slide, we have been discussing about what are hematinics, why should hematinics be given, what are the conditions of anemia, why is anemia caused. We had discussed about the types of conditions which leads to anemia. Then we have seen how iron is available in our living system. Next, we will learn about a very important component of blood, which is hemoglobin. Okay, we shall learn about hemoglobin. What is this hemoglobin? Hemoglobin is a protoporphyrin. It is a protoporphyrin. Each molecule of hemoglobin has 4 iron containing heme residues. Okay, each molecule of hemoglobin has 4 iron containing residues known as heme, which is bound to a protein called globin. Which is bound to a protein called globin. It has 0.33% iron. Hemoglobin has got 0.33% iron. Okay, this number we can understand when we move on to calculate what would happen if there is 100 ml loss of blood. See, I'll explain to you. If a human body loses 100 ml of blood containing 15 grams of hemoglobin, 100 ml of blood contains 15 grams of hemoglobin. This would lead to a loss of 50 mg of elemental iron, which is a huge number. It's a huge amount. Alright, now to raise the hemoglobin level in the blood by 1 gram per deciliter, our body requires 200 mg of iron. So now you understand what is the relationship between iron and hemoglobin. They are codependent. Okay. In order to increase 1 gram of hemoglobin in our blood, we require, or our body requires 200 mg of iron. 200 mg of iron is needed. Iron is stored only in ferric form. Iron is stored in our body in ferric form only. It is the active form of iron. It is present in combination with a large protein called apoferritin. Here we will see various sources of iron, or may I say various dietary sources of iron. The rich sources of iron include liver, egg yolk, oyster, dry beans, dry fruits, wheat germ and yeast. These are the rich sources of iron. Average sources include meat, chicken, fish, spinach, banana and apples. These are the average sources of iron. The meager sources include milk and its products and root vegetables. These are poor in iron content. Now we will move on towards our hematinic in general, which is ferrous gluconate. Ferrous gluconate. Ferrous gluconate has a chemical formula of C12H22FeO14. C12H22FeO14.2H2O. This is the chemical formula of ferrous gluconate. The chemical name of ferrous gluconate is, it is di-D-gluconate dihydrate, also known as ferrous gluconate. Gluconate dihydrate is another name for ferrous gluconate. It has got a molecular weight of 482.2 gram per mole. It has a molecular weight of 482.2 gram per mole. This belongs to a category of hematinics. As already mentioned, this belongs to a category of hematinics. Now, this is the molecular structure of ferrous gluconate. Molecular structure of ferrous gluconate. 6 carbon atom, 1, 2, 3, 4, 5 and 6. 6 carbon atoms attached to ferrous molecule, Fe2+. Ferrous gluconate is a greenish yellow to grey powder, slight caramel odor. It has got a slight caramelish odor. It tastes like caramel. It has a melting point, or it melts at 188 degrees Celsius. When we talk about solubility, it is freely but slowly soluble in water, giving a greenish brown solution, more readily soluble in hot water. It is practically insoluble in alcohol but soluble in glycerin. When we talk about preparation of ferrous gluconate, gluconic acid oxidation yields glucose oxidation yields gluconic acid. Okay. Ferrous gluconate is prepared in 2 steps. Number 1, oxidation of glucose. Okay. One molecule of glucose is oxidized to form gluconic acid. That means that is CH2OH, CHOH 4 times COOH. The aldehyde group of glucose is converted into acid, COOH, which is gluconic acid. This gluconic acid in the second step is treated with barium chloride solution, which is then treated with ferrous sulphate solution to yield ferrous gluconate. In this reaction, barium sulphate precipitates out and then is removed by filtration. Here if we see, ferrous sulphate is made to react with gluconic acid in the presence of barium to form ferrous gluconate. The barium is removed out in the form of barium sulphate. This barium is removed out as barium sulphate. This forms a precipitate which can be filtered out. The uses of ferrous gluconate include: it is used as a hematinic. It has less side effects when compared to other ferrous salts. The side effects includes constipation, diarrhea, nausea, vomiting, stomach pain, very common ones, loss of appetite, green colored stools, or temporary staining of teeth. The usual adult dose for iron deficiency anemia is: the initial dose is 960 mg per day of ferrous gluconate is given. Next, 120 mg per day, which produces or which provides 120 mg per day of elemental iron for 3 months. This is prescribed for 3 months. It's given in a divided dose of 1 to 3 times per day. Usual adult dose for vitamin and mineral supplementation is: initially 1 tablet is given orally for 3 to 4 times a day. Initial dose, that is 48 to 144 mg per day of ferrous gluconate is given, which contains 6 to 18 mg per day of elemental iron. So with this, we finish our topic of hematinics. Thank you.